This lecture will cover Chapter 14, The Principles of Disease and Epidemiology. Before we look in particular at a couple of epidemiological studies, I'd like to move us through some history of the study of infectious disease. And you'll notice that this history spans from about 400 BC all the way into the 1970s, so almost a 2,500-year-long period where different things happened that culminated in modern epidemiology. This timeline will allow us to highlight 6 of these. First up, we have Hippocrates, the Greek philosopher and physician who may sound familiar to you because of our discussions of prion diseases in chapter 13. And he lands here again because he was one of the first people to ever mention in writing that there was a material cause for disease. You might remember he dissected the brains of sheep and other cattle that had died and found their brains had changed drastically, and he attributed that change to a natural or material cause, in contrast to the view at that time, which was that the Greek gods were responsible for the good health and the bad health of both people and animals and all of the natural phenomena that people saw around them. About 600 years after the work of Hippocrates, Galen of Pergamon proposed something called the miasma theory of disease, and he used the word miasma because that's the ancient Greek word for pollution, and he was trying to make the connection that it wasn't angry gods that were cursing people with disease. There was something in the air, some pollutant in the air, a poisonous vapor perhaps, that got into a person and caused a disease. And all the way up in 1594, so 1800 years later, this idea of the miasma theory of disease was picked up by Girolamo Fracastoro, who proposed that the particles in the miasma were alive. So it wasn't a poisonous vapor, there were actually living particles in the bad air that were being moved into a person's body and causing disease. And that gets us to the second person on our epidemiology timeline, a London haberdasher called John Graunt, who was a hat maker, not an epidemiologist, but had a habit of recording very meticulously the birth rate, the death rate, and the disease rate of the people in his city, and so laid the groundwork for that modern retrospective collection of data that is so important in determining the causative agents of disease that we're going to talk about in a few minutes. And so even though he wasn't a scientist, we put him in here as an important contributor to epidemiology. Before we move forward to the next person on our list, just something to keep in mind, between 1662 and the 1800s, which is where we go next, we have some very important work that you might remember from earlier chapters, and that was the work done in 1674 by Antonin van Leeuwenhoek, who with his microscopes discovered animalcules. Remember, he even discovered bacteria. He was able to see bacteria, different protozoans, and what he found was that these animalcules were everywhere. And if you think about that in connection with the work of Fracastoro proposing that the particles in miasma are alive, you can see how this is all starting to culminate in what we now know as the infectious transfer of disease. And in fact, in 1844, Agostino Bassi actually identified a microscopic fungus on sick silkworms and found that they were not present on silkworms that were not sick, and started making this connection again. So this stuff starts happening, and then we move to 1858 with a name that probably sounds the most familiar to you of all the names on this list, and that is the name Florence Nightingale, who of course is the founder of nursing. But here she's known because as a nurse during the Crimean War, she worked in a British hospital where 10 times the number of people were dying of infectious disease as were dying of battle wounds. And she started looking around for causes of this infectious disease outbreak, and she saw bedpans that weren't being emptied and were being left by patients. She saw sheets that were never being changed, surgical dressings that were never being changed, and she proposed a major change in sanitation in the hospital, and she actually managed to drastically reduce the infection rate there. So well known for being a nurse and the incredible care that she gave to patients, but also for sanitizing hospitals. Next, we have 1865, and another name that should sound familiar to you, Joseph Lister, of course, the discoverer and marketer of Listerine, which cures all of your ails when it comes to bad breath. But what we know him best for here is that he was one of the first people to propose using a chemical to clean surgical wounds and surgical instruments prior to surgeries. And he did this because he was reading the work of Louis Pasteur and understanding that there were microorganisms present that could be used to inoculate other things. And he thought, hey, maybe we should start sanitizing our surgical instruments in order to prevent the spread of infection. And so he gets a nod here. About 10 years later, we pit stop with Robert Koch, who is famous for lots of different things, infamous for some things, like thinking he found a cure to tuberculosis when he really hadn't. But what he's remembered most for is Koch's postulates and the germ theory of disease, which we will spend lots of time talking about in the future. So put a pin in that one and you'll see his name in a few more minutes. We're going to wrap up the history of the study of infectious disease with some applause for healthcare workers all over the world who in the 1970s worked hard and eradicated the world of smallpox. The last known smallpox case that occurred naturally was in 1977 in Somalia, Africa, and it was declared eradicated in 1979. And the success of that work actually led to other eradication campaigns for polio, which has almost been eradicated from the world, and also the guinea worm, Dracunculus melionasis, which we mentioned in our diversity unit, which is close to eradication as well. Remember, when we discuss timelines, I'm not so concerned that you remember exact dates. What I would like you to know is the flow of information. So what came first, what came after that, because knowledge is cumulative. And so looking at what people learned and what that contributed to the next person's understanding of the process is important. Before we start talking about true pathogens, we want to take a moment to remember that not all microorganisms interact with human beings in a negative way. There are many symbiotic relationships that exist between human beings and microorganisms, and not all of them are detrimental to the human. In this figure, you see three kinds of symbiotic relationships. Commensalism, where one organism benefits and the other is unaffected. An example that your book gives of a commensal relationship between two organisms would be the relationship between Staphylococcus epidermidis and human beings. Now, I would argue that Staphylococcus epidermidis actually does provide a benefit to the host because its production of acids and its very presence on the skin would make it more difficult for would-be pathogens to live on the skin. But I think what's important here is just to understand that the idea of a commensal relationship is one in which one organism benefits and the other is unaffected. Now, what I was just describing to you would be considered a mutualistic relationship where both organisms benefit. And the example that your book gives here is completely accurate, and that is the relationship between you and the E. coli that live in your large intestine. You might remember our discussions in chapter 1 where we talked about normal flora and how those microorganisms, particularly in the intestinal tract, are so important with their production of growth factors and vitamins and their ability to increase your water absorption from your colon. And again, just because their very presence there makes it less likely that would-be pathogens could take hold in the intestinal tract. Now, those are both relationships that we would consider good, and those are both relationships that we have with our normal flora. In example C, we have a relationship where we have a true pathogen, a parasite, right? Where one organism benefits, that organism would be the pathogen or the parasite, and the other is harmed by the relationship, and that would be you, the host. Now, most of the time when we think about parasites, we think about organisms that are all the time causing infection. Now, there are things called opportunistic parasites, which only will cause damage to the host at particular times or in particular circumstances. One which is not that kind of opportunistic pathogen would be the H1N1 influenza virus, shown here in orange on a host cell, which is shown in green. As promised, before we move into modern epidemiology, we want to revisit the miasma theory of disease, which was first proposed in 200 AD, with that ancient Greek word for pollution, miasma, being used to describe a poisonous vapor and not the angry gods, which was causing disease. That was the reigning scientific theory even into the 1600s. You'll remember from our timeline that in 1594, Girolamo Fracastoro proposed that the particles in miasma were alive. So in the 1600s, in the 1700s, in the 1800s even, there was still this miasma theory of disease. There's bad air causing disease. And we want to leave that there for us as we move into talking about the modern understanding of the transmission of disease, which is attributed to the work of Robert Koch, who came up with Koch's postulates, which are today the criteria that are set forth to connect a causative agent to the disease that it causes. And so instead of the miasma theory of disease, we have the germ theory of disease. So the germ theory of disease results from the very careful work done by Robert Koch in the 1800s, where he connected a particular microorganism through a series of postulates and experimentation to a disease. So if we look at Koch's postulates stepwise, we find that step number one is that microorganisms must be isolated from the diseased or dead animal. Seems like a simple step, but that's where we begin, right? The simplest experiments are the ones that end up making the most sense to people and are the ones that end up holding on in the long run. Step two is that the microorganisms from that diseased or dead animal must be grown in pure culture. So you see there are colonies resulting from the extraction of a sample from that mouse, and then those colonies being grown out and isolated on petri dishes. The second half of step two is the identification of those microorganisms which have been grown in pure culture. Once those organisms have been identified and named, then you can move on to step three. Step three involves the inoculation of those identified microorganisms into a healthy laboratory animal to see what happens when a new animal has been exposed to that possible etiologic agent. Step four requires that the same disease can be reproduced in that second laboratory animal. So you see there that the microorganism that was isolated and identified from the mouse in step one has now caused the same disease in that second laboratory animal. Now you might think that you're done at that point, but Robert Koch was a very careful man, and so he didn't stop at step four. He went on to step five. And step five requires that those microorganisms must be isolated from the second animal and again grown in pure culture so that the microorganisms can be identified again. And finally, Koch's postulates and the confirmation that the microorganism from the first diseased host has caused the same disease in a laboratory host. And once you can satisfy all five of Koch's postulates, you are able to say experimentally what the etiologic or causative agent is of that particular disease. This work, as I said, was incredibly important because it made a connection between particular microorganisms and a particular disease process. Before we move on to modern epidemiology, I'd like to take a few minutes to talk about two really cool discoveries that were made in the 1800s prior to the work of the germ theory of disease, which I think makes these even more amazing because this work was done without people understanding the clear connection between microorganisms and disease and without those postulates that they could rely on in order to make that connection. So the first of those is the work of Ignaz Semmelweis, who was the chief resident of a hospital in Vienna from 1846 to 1848, and his claim to fame is that today he is known as the savior of mothers because he reduced the incidence of something called puerperal fever or childbirth fever, which is a fever that is contracted by a mother soon after childbirth. So it's not an infection of babies, it's an infection of mothers, and it happens as a result of them giving birth. So in Vienna at that time, there were two big clinics, and one of them was a teaching hospital, and the second clinic, which was not a teaching hospital, was staffed primarily by midwives. Both of those places had obstetric wards where women could come in and give birth, but Clinic 1 had such a reputation for puerperal fever that women frightened of giving birth inside the hospital would actually give birth at home in order to avoid going to Clinic 1, which meant that Clinic 2 was far more crowded. And this was a problem that people in the hospital administration were trying to figure out. And completely coincidentally, Ignaz Semmelweis has a friend who is performing an autopsy with a medical student and accidentally gets stabbed with the scalpel, and he contracts a disease which looks just like the victims of childbed fever or puerperal fever. So Semmelweis is looking at this and he hypothesizes, because again, there's no germ theory of disease, that doesn't happen until the 1870s, that there were cadaverous particles that had contaminated the wound. So he starts thinking about this, and he starts thinking about what medical students do in their off time, and most of the time what they were doing was going and moving through these autopsies in order to learn the human body, and they weren't washing their hands afterwards. So Ignaz Semmelweis came up with what may seem like a very obvious solution today. He made them wash their hands with a bleach solution, and the incidence of childbed fever or childbirth fever dropped by 90%, and thus Ignaz Semmelweis was coined the savior of mothers. So about the same time that Ignaz Semmelweis was saving mothers in Vienna, John Snow was concerned with saving people of all ages from cholera in Soho, London. Now, in the 1850s, there was an epidemic of cholera in London, which killed about 31,000 people, and at its height was very, very deadly. There was a period of time where within 15 days, 616 people had died. Within that 15-day period, there was a 3-day period where 127 people died in the Broad Street neighborhood of Soho, and many of them were treated by John Snow. So he decided to try to figure out what was causing these people in the Broad Street area to have such a high incidence of this disease. So he very meticulously went around with a map of Soho, London, and the Broad Street area and went to every house and recorded how many people had contracted cholera. And you can see each of those cases of cholera in the map on your screen. All of those red dots are cases of cholera. And what he found is what might be very obvious to you. That blue dot right in the middle there is the Broad Street water pump. So he started thinking maybe this was a waterborne illness, and maybe the Broad Street water pump was what was transmitting this disease to everybody. So he moved around and started interviewing people, and he found some outliers to his hypothesis. One of them was a workhouse slash prison in Soho that had 535 inmates and almost no cases of cholera. So he went and asked them, and it turned out they had their own well and they bought water from a waterworks. So they didn't have any cholera because they weren't using the Broad Street pump. He confirmed this hypothesis by interviewing workers in a brewery on Broad Street who again confirmed that they had their own well water and they were far more likely to be drinking beer during the day than they were water anyway. And to compound that and all of the data from the community, there was a factory near the pump which used its water and 16 of its workers died. Now there was one really strange outlier, and that was a woman and her niece who lived really far away from Broad Street who had also died of cholera. So in order to try to figure out what was going on there, Jon Snow went and interviewed her, the woman's son, and he said, oh yeah, she used to live on Broad Street, loved the water, and had bottles of it brought to her house. And in fact, she had shared a bottle with her niece, and they both died the following day. So Jon Snow figured this out, and he actually went and removed the pump handle so that people just couldn't get water from the Broad Street pump. And lo and behold, the incidence of cholera in the Broad Street area drastically was reduced. So he published his data in a big long treatise on cholera, which you can actually find online. It's very long. I don't know that you'd want to read it, but you certainly can. You can't go visit the pump that he removed the handle from because it has been removed, but you can go visit a replica, and you can visit the Jon Snow pub in Soho, London, should you so choose. So what we've been talking about are different epidemiological studies, which begin with something called a descriptive epidemiological study, where you collect and analyze data. So if we talk about Jon Snow, which is a great example of this, he goes out, he interviews people, he records meticulously all of the incidents of cholera in a particular area, and he tries to determine the commonalities amongst those people. And if you think about that, right, that idea comes from the work of John Graunt in the 1600s, who meticulously recorded the birth, death, and disease rates in London during his time. And his doing that is picked up by other people like Jon Snow, who uses that here to perform an experiment where he tries to now control the transmission of the infectious disease. And so he does first this descriptive study where he collects data and analyzes them. He comes up with a hypothesis. His hypothesis, which is meant to stop the transmission of the disease, is that the water coming up through the Broad Street pump is contaminated. He performs an experiment in order to test that hypothesis. Now, he's not going to perform a controlled experiment where he has a control group and an experimental group. He's not going to feed people water he thinks is contaminated with cholera and then feed people water from somewhere else and see what happens. What he's done is really an analytical study where he's looked at diseased and healthy people and tried to determine what's the difference between them, right? And what does he do? He just removes the handle of the pump. Doesn't do a controlled study, just removes the pump handle, which he thinks is the cause, and says, let's see how things go from there. And then he analyzes his data. Did the transmission of the disease stop? In the case of the cholera epidemic, yes, it did. And so that worked. And it actually turned out that the entire Broad Street epidemic was traced back to a baby who had contracted cholera. The stool from the diapers was actually buried under the house, and it made its way into the groundwater because in the 1800s in Soho, remember, there's no sanitation, there's no sewage system, there's also no water pipes, so you're getting groundwater. And with no bathrooms, the common thing to do was to dig holes in the dirt floor of your house, bury your waste, your fecal material, and your urine, and just put dirt over the top of it. And when you do that, of course, it can trickle into the groundwater. And cholera, it turns out, can persist in the environment for extended periods of time. So Jon Snow's work there really was not one of these three. It involved all of this. He went through and did a descriptive study. he compared diseased and healthy groups, and then he devised an experiment to test that hypothesis. Had he been wrong, of course, he would go back to the drawing board and determine another hypothesis, just like you do with every other aspect of the scientific method. All right, I'm sure you've had enough history, so we are going to fast forward now to the 21st century. To a norovirus outbreak in 2005 in Kent County, Michigan. Norovirus, just for background information, is a gastrointestinal pathogen that causes diarrhea and vomiting. And what we're looking at here is an outbreak of norovirus, which began on the 5th of May in 2005 in a group of people who had all eaten together at a school luncheon on May 3rd. And right about the same time this is happening, there was a luncheon on May 5th at a publishing company, which resulted in several people contracting norovirus on May 6th. Likewise, on May 6th, there was a gastrointestinal illness outbreak in a social service group who held a luncheon on the 4th of May. All three of those places contacted the health department and let them know that all of these people had eaten the same meal and contracted the same illness. And so they got the health department involved in this. And as the health department started researching it, they found community cases starting on May 4th. And so the health inspector went out to the place of business where all of these meals had been served, which happened to have been a franchise submarine sandwich restaurant. And they noted that this restaurant had served all of the meals, and so they requested that the restaurant do a thorough cleaning. The restaurant did that thorough cleaning on the 7th of May, and because the incidents of this norovirus were still occurring, the restaurant voluntarily closed on the 10th of May in order to be cleaned by a professional cleaning company. Now, in the meantime, the health department was collecting stool samples from all of these people who had gotten sick, and by the 16th of May, they confirmed that those cases were all caused by the same strain of norovirus, which was also found in a seemingly healthy food handler who worked at this submarine sandwich shop. So on the 16th, they sat down with this food handler and asked him or her what was going on, and that food handler reported that on the 2nd of May, he had been ill and missed work, but had come back to work on the 3rd of May, and had done that because he had had 12 hours of no vomiting and so felt himself safe to come back to work. Now, I know what you're thinking. You're thinking this guy came back to work and he didn't wash his hands, but you're wrong. He did wash his hands. The problem is that he washed his hands in the food prep sink, right? So you might have noticed if you go to restaurants that have sort of an open kitchen that there are signs on the sinks that say, you know, for hand washing only, for food preparation only. And the reason for that is when you wash your hands, you wash the microorganisms off of your hands and into the sink. And if someone comes and preps food like, say, lettuce in that sink, then the lettuce will collect the microorganisms and then will be placed on a sandwich where that microorganism gets a straight shot into the gastrointestinal tract of its next victim. And that's exactly what happened here. It turned out when they interviewed all of these people who had been sick, that all of them had lettuce on the sandwiches that they ate from the submarine sandwich shop. So once they've identified the source of the contamination, they on the 17th professionally cleaned the restaurant and it opened its doors on the 18th of May. So problem solved there. Now we're going to think about what do we do with these data, right? How did they know to go back to this place where all of these people had eaten, right? Why wouldn't they instead just think that, like many times, this norovirus was just kind of making its way through the population? Well, the reason they knew to go back to this submarine sandwich shop is shown in this graph here, where you see very few norovirus infections, and then a massive upswing in norovirus infections, and then a quick drop in the number of norovirus infections, because that graph looks very much like the red line on this graph to the left. And when a series of infections looks like that kind of graph, we call that a common source epidemic. Those data are indicative of a common source for that epidemic. In this case, that common source was the submarine sandwich shop. And that would be in contrast to a host-to-host epidemic, which is shown here in green, where you have a few cases, and then slowly over time the number of cases increases, and then slowly over time the number of cases decreases again. So understanding the difference between what a common source epidemic looks like and what a host-to-host epidemic looks like is really important, because we have very different mechanisms for controlling that disease transmission based on how it's being transmitted from person to person. Now, if it's a common source, go to that common source, clean it, shut it down, do whatever you have to do to stop that source from contaminating other people. If it's a host-to-host epidemic, even if it's cholera, it's a very different strategy. In a host-to-host epidemic, you find through education of the public, you're able to decrease the incidence of disease, right? Explaining to people the importance of washing their hands after they use the restroom, the importance of maintaining cleanliness in their food and things like that, right? So looking at the data and determining what kind of epidemic is important, right? Because again, the goal of epidemiology is to stop the transmission of disease. Before we move on to really the main topic of chapter 14, which is understanding infectious disease, I want to stop for a moment and introduce you to some terminology that's going to be used throughout chapter 14, 15, 16, 17, 18, all the way through the rest of the semester. And the first of those words is one that should be familiar to you already, and that is epidemiology, which is the study of disease transmission, which of course in this class we study the transmission of infectious disease, which always begins with an index case, which is the first patient who has contracted the disease during an outbreak. That person will sometimes also be called patient zero. When we're determining who patient zero is, one of the major things that's examined about people who have gotten sick is the pathology of the disease, how the disease progresses through a particular host, what the severity looks like, and then tracking back to the person who first showed signs of that disease. Once you've identified patient zero through that pathological study, your next step is to determine the etiologic agent of disease, or the causative agent of disease, right? What is the organism that is causing that disease in that person? Now that leads us to two words that you may think you know the definition to, but we want to make sure that you know the epidemiological definition to. The first of those is infection. So infection is a word we use often, but in this case, in epidemiology, infection means that you have been colonized by pathogens, right? So in this case, colonized by the etiologic agent. And when you have been colonized by the etiologic agent, oftentimes, if you are not an asymptomatic carrier, that will lead to a disease state, which is the abnormal body function, which is associated with the disease. As you try to make sense of the disease, you study what's called the pathogenicity of the disease, and that is where you look at the signs and symptoms of the disease, which are often abbreviated S and S. S1 for signs, which are the outward measurable aspects of disease, like fever, inflammation, things that can be noted, high blood pressure, low blood pressure, tachypnea, apnea, things like that that can be measured. The other S, the second S in S and S, are the symptoms of disease, and symptoms and signs are not the same thing. Signs are outward measurable things, symptoms are how you feel, and we have a bad habit, I think, of discounting how you feel versus what kind of signs you're exhibiting, right? What's that moms always do to their kids? Well, do you have a fever? You don't have a fever, go to school, right? How do we know we're safe to go back to school? If you've been fever-free or you've stopped vomiting or you've stopped having diarrhea for the last 24 hours. But anyone who has ever had a sick infant or a sick person with dementia in their life, and that person has not been able to communicate how that disease makes them feel, it can be really difficult to identify what actually is happening in a person. So we don't want to discount symptoms. Symptoms are important. Aches and pains and nausea and things like that can be just as important to a diagnosis as a fever or a change in heart rate, blood pressure, or breathing. So once you have identified the etiologic agent of disease, you have to decide whether or not that agent is actually infectious, which sounds weird because how do you have an infectious agent that is not infectious? But hold on, that actually can happen. So infectivity tells us how infectious a particular microorganism is. And to say that a microorganism is transmissible from one person to another through some means is to say that it is communicable. It is able to be communicated between one person and another. If that agent is very easy to transmit from one person to another, we call that agent contagious. If that microorganism can't be communicated from one person to another, we call it non-communicable. Non-communicable infections typically are caused by opportunistic normal flora, like a urinary tract infection, or introduced bacteria, like a IV stick in a hospital, which contaminates the bloodstream and causes septicemia. And they're not considered communicable because they're inside of the body of the person that has contracted them and won't be passed to anyone else. And that, though it is a lot, is the last of the definitions that I have for you right now. So if you're one of those people who enjoys making flashcards, have fun because you've got lots of fodder for flashcards here. And keep all of these terms in mind as we move further through chapter 14 and through the rest of this lecture unit. And now, finally, we get to understanding how infectious diseases work. And again, we want to keep in mind that when we identify infectious diseases by using these 5 processes, we're always looking to stop the transmission of disease. So we're going to look at 5 aspects of infectious diseases. We're going to see how those 5 aspects can be used to classify diseases, look for patterns in disease, and look for ways that diseases are spread, again with the overall goal always of stopping the transmission of infectious diseases. So we'll look at 5 things. We'll look at the progression of a disease. What does it look like in the host? We'll look at the occurrence of disease. What does it look like in the community? We'll look at the severity and duration of the disease. How does it run its course in a particular person? We'll look at the extent of host involvement, how much of the body is affected by this particular microorganism. And then we'll look finally at the transmission of the disease. How is the disease communicated from one person to another? Though diseases move through an individual at varying speeds and with varying severity, there is a rhyme or reason to the way they move through a particular person, and that is called disease progression. Disease progression, as you can see from this chart, has to do with the amount of time and the number of microorganisms that a person has been infected with. For every microbial illness, there is what's called an incubation period, a period of time where there are no signs or symptoms and there are very few microorganisms present in the person's body. As those microorganisms begin to reproduce, whether they are viruses or protozoans or bacteria, they will increase in number, and as they increase in number, the number of symptoms or the severity of symptoms will increase as well, and the person will move from the incubation period where there are no signs or symptoms to what's called the prodromal period, which is Greek for precursor, where the person exhibits mild signs or symptoms relative to what will be full-blown illness. As that's happening, the person's body defenses are being overwhelmed, the microorganism or virus is continuing to reproduce, increasing in number, and that just increases the signs and symptoms of the disease and moves from a prodromal period into a period of illness where you have ever increasing signs and symptoms, ever increasing number of microorganisms, and then you hit this peak point, which is sometimes called the nadir of disease, where you feel the absolute worst. The microorganism is doing its absolute best, and you feel the absolute worst that you are going to feel through this illness. And then either because of intervention medically through the use of antibiotics or antivirals or some other therapy, or simply because your immune system starts to take hold and recover, the number of microorganisms starts to decline over time, and you enter what's called a period of decline. And through the period of decline, your signs and symptoms move from the maximum down to a level that mimics the prodromal period. And the difference here is that you're getting better over time until you hit a point where you have very mild signs and symptoms, which move to no signs and symptoms. But here you're no longer considered to be in the incubation period. You're considered to be in the period of convalescence, where your body is recovering from the illness and you are ridding the body of microbes. Unless, of course, you're entering into what we discussed in chapter 15 would be a latent disease or something of that nature. So what we're describing here is really an acute illness, not talking about whether it's going to be chronic or latent or anything like that. And what we want to get from this is the number of microbes in the body has an absolute effect on how you feel. So the progression of disease through the body has everything to do with the number of microorganisms in the body at a given time. The second aspect of understanding infectious disease that we're going to talk about is occurrence of disease, which answers the question, what does the disease process look like in the community? How many cases of the disease are there in the community? And more important than that, do we expect that number of cases, or does that exceed the expectation that we have for that particular disease? So an endemic disease is one that occurs at a low, constant, expected rate. For instance, we have cold and flu season, where we expect to see incidence of cold and flu increase during the winter months. That's not unusual, that's not unexpected, but of course, when things become greater than expected, when we have more cases than we expect to have, then those things become epidemic. Epidemic, which is shown in the third diagram here, is where you have more than the expected number of cases in a particular area. And you can have an epidemic in San Diego County, you can have an epidemic in California, you can have an epidemic in the United States, you can have an epidemic in North or South America. So it doesn't necessarily tell you how big the area is. What it says is we have a higher than expected number of cases of a particular disease. So endemic diseases can become epidemic diseases. Another kind of a disease that can become an epidemic disease is a sporadic disease, and sporadic diseases, unlike endemic diseases, are not consistently found in the population. They sort of crop up and they go away. And whereas flu would be an example of an endemic disease, Ebola would be something that's considered sporadic. Ebola crops up in a limited area in Africa, and because it's so highly contagious, it spreads very quickly in that area and goes from sporadic to epidemic. So either an endemic disease or a sporadic disease can become epidemic, because epidemic, again, doesn't talk about the severity in numbers or even in symptoms. It talks about whether or not you've exceeded the expectation for the number of diseases in a particular area. Now, the problem with any of these situations is that in the modern world, we travel so quickly from one area to another that it's very easy for something that is epidemic to become pandemic. And that happens because, say, a person in California has a disease They're in the incubation period, so they have no idea that they have the disease. They are not really showing any signs or symptoms of the disease, but they may be able to transmit that disease even on the airplane. But the problem is when they get off the plane, say in Italy, they now have the disease and they've now moved it to a different continent, and now it can become epidemic in Italy. So in this point, you have an epidemic in California, an epidemic in Italy, and you have a pandemic. You have a higher than usual number of cases on more than one continent. So it's all about the numbers. Are the numbers what you expect to have? Are the numbers exceeding the expectation that you have? And then is that happening on more than one continent? One of the things that will determine whether or not a disease is sporadic or endemic or becomes epidemic is something called herd immunity. And herd immunity is a measure of how many individuals in a population are resistant to a disease. So in this population here, you have a single person. So imagine this is the person that hopped on a plane in California and flew to Italy, and they are infected, and they get off the plane in Italy, and no one in Italy has ever had that disease, nor have they ever been immunized against that disease, so they are all susceptible. And if that person is capable of infecting people, that will very quickly become epidemic. As you can see from this diagram here, one person infects 2, those 2 infect 4, those 4 infect 8, and that epidemic just grows and grows and grows. And what was sporadic in California is now epidemic in Italy. What we can do to avoid this is have a level of herd immunity, which is shown in this diagram here, where we have blue individuals in the population. Those are people who have either contracted the disease or have been immunized against it. Either way, they are immune to the disease and they block transmission of the disease. So now that person gets off the plane in Italy and they expose the first 2 people to infection. One of them gets the disease, the other, notice, remains blue because that person is immune to the disease, which means that that person does not spread that disease anywhere else. So we have one person infecting one person, infecting one person, infecting one person, and that allows us to keep that disease from becoming epidemic. Today, most of the way that people get herd immunity is through immunizations. In the past, before immunizations were common, herd immunity still occurred, but it occurred when a disease moved through a population and either killed or made everyone there immune to that disease. And there are really interesting data, especially as we backtrack and look at influenza pandemics. And it's thought that the influenza pandemic would move through a population like the one shown at the top here. Everyone would contract influenza, and people would either die of influenza or they would recover and be immune. And then you would have a situation like we have in the bottom here, where at least half of the population is immune to the disease. And so the disease can't move through the population and cause a pandemic. But as those people, the blue and the black individuals, age and die in the population, they are replaced by more black individuals who are not resistant to the disease. And so you see these cycles of infection that correspond roughly to generational gaps in time. So a resistant population survives the influenza pandemic, and they don't have any pandemics for a few decades, several decades, and then those people are slowly replaced in the population by susceptible individuals, and then the endemic incidence of influenza becomes epidemic and pandemic again. So today, our major public health move toward herd immunity and keeping diseases from becoming epidemic is immunity through immunizations. All right, now we're going to talk about the third aspect of disease classification, severity. Severity looks to answer the question, how does a particular disease run its course? This is more closely related to disease progression because when we talk about disease severity, we're talking about the course of the disease in a particular person. Disease severity is characterized by both the progression and the duration of disease, and it ranges from acute to chronic to subacute to latent and to persistent based on how the disease progresses through a host and how long a host is affected by a disease. So the progression and the duration, you can imagine, have a lot to do with that previous slide on disease progression. And this sort of gives you an idea of the differences in the timeline of disease progression between incubation, prodromal illness period, and the decline in convalescence as a person recovers from disease. Acute diseases are characterized by a rapid progression and a short duration. So a person moves very quickly into the period of illness and then very quickly into a period of convalescence. Influenza or the common cold would both be examples of acute diseases. On the other hand, you have chronic diseases that have a very slow progression and a very long duration, and perhaps that duration never actually enters into a period of convalescence. A lot of times chronic diseases are diseases that persist through a person's lifetime. So these diseases would really drag out that timeline that we saw in the disease progression slide. Subacute diseases tend to be slower than acute, but faster than chronic. So you can imagine there's this large continuum, a lot of gray area between acute diseases on one end and chronic diseases on the other. In addition to subacute diseases, we have both latent and persistent diseases. Latent and persistent diseases may seem similar, but the difference between the two of them is that during a latent disease, there are periods of inactivity, apparent inactivity, where there are no signs and symptoms of disease, whereas persistent diseases have symptoms which flux between acute and chronic, but there is always some disease syndrome present. Now, most diseases have a particular severity. They are either acute, chronic, subacute, latent, or persistent, and you can see each of these categories has a disease associated with it. So influenza is typically acute, leprosy is typically chronic, measles are subacute, herpes is a latent disease, and gingivitis is an example of a persistent disease. But all of those things depend on the predisposing factors that are affecting a particular patient, and any predisposing factor actually will increase the severity of the disease. So maybe something will never be latent, it will never have a period of inactivity, or a persistent disease will always tend toward the acute portion of disease, and maybe a chronic disease or a subacute disease will look more acute in a person who has a predisposing factor. And the three major predisposing factors are gender. Females tend to be predisposed to an increased severity of disease, particularly females who are of reproductive age, because ovulation tends to decrease the immune system temporarily. Age is another predisposing factor. Not so much young people. Young people, babies have exceptional immune systems, just that they have immune systems which have not really encountered a lot of foreign entities. Really, when we talk about age, we're talking about the upper end of age. So as people get older, their immune systems don't function as well, and so old age is considered a predisposing factor. And then, of course, immune or genetic status. So some people genetically have a stronger immune system than others. Some people have a more robust immune system. Obviously, a person who's on immunosuppressant drugs or has an immune system failure or infection are going to be more predisposed to a more severe disease. Next, we have host involvement, which seeks to answer the question, how much of the body is affected by the disease? And initially, when we talk about that, we talk just about the location in the body that's affected. So the very most simple explanation for host involvement would be a local infection, and that is one where just in one area of the body you have a local infectious process, local disease syndrome, and that could be something as simple as an infected cut or an abscess. Of course, the problem with localized infections is that because your entire body is connected through your cardiovascular and lymphatic systems, that once a pathogen gets into either of those two systemic systems, that local infection can very quickly become systemic. So a systemic infection is one which is in the bloodstream. Now, depending on the pathogen, systemic infections are called different things. So if you look at the left-hand side of this diagram, you'll see a picture of systemic infection, and below that diagram we have the words bacteremia, viremia, toxemia, and septicemia. Notice each of them ends in the word emia. Emia is a word that refers to blood, so bacteremia is the presence of bacteria in the blood. Viremia is the presence of viruses in the blood. Toxemia is the presence of toxins in the blood. And septicemia is actually growth in the blood. Remember from our discussions of aseptic technique that aseptic technique is without growth. So septe means growth, so septicemia means the growth of pathogens in the blood. Now, we make a distinction here between bacteremia and septicemia, saying that bacteremia is the presence of bacteria in the blood and septicemia is the growth of bacteria in the blood. Clinically, you won't oftentimes see that distinction being made. You'll a lot of times hear people talking about bacteremia when what they actually mean is that there are bacteria growing in the blood. For the microbiologist, the distinction is important because it allows us to understand whether or not bacteria have just been introduced into the bloodstream because of an injection or an IV catheterization, or if a person actually has a systemic infection. So we make that distinction here in this class, but a lot of times clinically that distinction is not made. Now next, kind of right in the middle of the slide, you see something called a focal infection. And hopefully you can tell from the slide that there is a focus. There's actually an arrow drawn from the nasopharynx to the heart. And what that's trying to signify to you is that there are bacteria that cause infection in the naso and oropharynx, which will then focus their attention to a particular part of the body, in this case, the heart. And that's actually a bacterium that we've talked about before. I mentioned streptococcal bacteria, Streptococcus pyogenes, to you a couple of times, and the fact that that can be a very dangerous organism to become colonized by, because they will move from an upper respiratory tract infection of the pharynx if they get into the body, and they will move directly to the heart where they will damage heart valves. So they don't tend to be systemic, they tend to go straight from that respiratory tract into the heart. They focus their attention there, and so we call them a focal infection. Further on to the right, we have what's called a subclinical infection, and subclinical infections, very importantly, have no visible signs and symptoms and may be caused by various microorganisms. So there might be a little bit of general discomfort or malaise, but a subclinical infection is one where, though there is colonization of the body by a pathogen, there's no apparent signs and symptoms. And then finally, all the way over on the right, we have two kinds of infections that are always associated with one another, and we call them primary and secondary infections. As their names suggest, primary infections occur first, and secondary infections happen as a result of the primary infection. The example that's given here is a urinary tract infection, right? Urinary tract infections in females are often treated with antibiotics, which will then, because they're more broad spectrum, clear out a lot of the bacterial normal flora of the external genitalia of the female host, and that will leave them susceptible to a secondary yeast infection of the external genitalia, and so we call that secondary vaginal infection, secondary because it's caused by the treatment of the first. Now, secondary infections are not always caused by the treatment of the primary infection. Sometimes secondary infections happen just as a result of the body being worn down by the primary infection. So an example of this that would be equal in both males and females would be a respiratory tract infection where a patient has a primary viral infection, and that viral infection kind of beats up the respiratory tract, wears down the immune system and the body defenses, which leaves a person susceptible to a secondary bacterial infection, which would not have been able to take hold if the viral infection had not first weakened the host. Next, we have disease transmission, which seeks to answer the question, how is a particular disease communicated? How is it transmitted from one person to another? There are three basic mechanisms of transmission between hosts, and they are contact transmission, vehicle transmission, and vector transmission. As you can see, within each of these three categories, we have subcategories of transmission. In contact transmission, you can have either direct contact, indirect fomite contact, or indirect droplet contact. So in contact transmission, there's the contact means that you have a host who comes into either direct or indirect contact with a susceptible individual. So direct contact means that there's actually a direct contact, physical contact between the infected individual and the susceptible individual. So any disease that happens as a result of direct contact between two people, such as any of the sexually transmitted diseases, would be considered a direct contact transmission. Indirect fomite contact transmission happens where an infected individual accidentally or intentionally deposits the infectious agent on a non-living object, and then a susceptible individual touches that object and then contracts the disease from them. Indirect droplet contact happens when the droplets that are either sneezed or coughed from one person because they contain microorganism and then land on another person cause that second person to contract the illness. So the common cold is a great example of a contact transmitted disease, and it actually can be contracted directly if a person who has a common cold, say in their nose, were to touch noses with another person. That would move the virus directly from one person's nose to another, which would be an example of direct contact. Also, a person with a common cold might sneeze onto a telephone or remote control or something like that, which a susceptible person could then touch and move that virus onto their hand. And then if they were to itch their nose, they would move that virus into their nose, and that would be indirect fomite contact. The third way that you could contract rhinovirus would be if someone sneezed rhinovirus right onto your face. As gross as that sounds, the droplets would contain the virus and they would land on your nose, and that would allow them to get to you indirectly from that infected individual. The next way that diseases can be transmitted is through a vehicle, and vehicles are always water, air, or food. So water is when the infectious agent, the virus or bacterium or whatever it is in water, which is then drunk. Food obviously happens when the food has been contaminated. And airborne, though it looks like it's very similar to droplet contact, is not the same. Airborne are things that can stay in the air for a distance larger than a meter. So these are things that could be contracted across the room rather than, say, a person sneezing on you and having to be in close contact with you. So vehicle transmission is always water, air, or foodborne. And then finally we have vector transmission. Vector transmission always, in this case, has to do with bugs, right? Because remember, a vector, we learned in chapter 8 and 9, is something that moves something from one place to another. In chapters 8 and 9, we were talking about moving DNA. Today we're talking about moving pathogens. So we have on the left-hand side here the mechanical vector. A mechanical vector simply mechanically moves the pathogen from one place to another. So here you have a picture of a fly who's landed in the trash can. Assumably he's picked up microorganisms in the trash, and then when he lands on the sandwich, those microorganisms fall off of his feet and onto the sandwich. And then when you eat the sandwich, you then contract the illness. On the other side there, we have the biological vector. Biological vectors transmit pathogens when they bite a host. So you can see here in this picture, this is a tick. So presumably the tick has bitten the rat that's lying on the ground there, pulled the microorganism out of the rat when he bit it because it was a blood-borne pathogen, and then when the tick bites the person, he transmits that disease to the person. So when you think about this, you can think about biological, the pathogen's actually living inside of the bug that's transmitting the disease, right? And the reason I don't say insect is because technically speaking, ticks and spiders are not insects, they're arachnids. So we would have to say arthropod, which is kind of an odd word. So we'll just say bug. So vector transmission is always bugs, either mechanically or biologically. All right, so let's see how these 5 aspects of disease classification might work together to help to diagnose, treat, and stop the transmission of a particular disease. So to do this, we'll look at 2 things, the common cold and influenza, and we'll look at how those things are different in each of these 5 categories in order to understand how we can differentiate between the 2. So first of all, occurrence. So common cold, about 1 billion cases of common cold per year. Influenza, about 35 million cases a year. So when a person shows up and they have symptoms of an upper respiratory tract infection, probably the clinician is thinking common cold more than influenza because common cold is more common than influenza. In terms of severity, as the person comes in and they start talking about what's going on with them, if they have both of these would present as acute diseases, though the common cold would present more gradually than influenza. So the movement from prodromal into illness would be much faster with influenza than with the common cold, right? Maybe a day for influenza and a couple of days to common cold. So when they ask you, how long have you been having these symptoms? This is what they're trying to figure out, right? Whether this is something that's a gradual acute or an abrupt acute. And then they're going to ask you what's going on, how you're doing, right? So if you have a common cold, you're more likely to have only upper respiratory tract infection symptoms like runny nose, stuffy nose, things like that. If you have influenza, you probably have upper respiratory and lower respiratory tract symptoms. So you're going to have the nose, throat, and then you probably have whole body effects like fever, chills, headache, maybe your ears are involved, ears are popping, ears are sore, body aches, and you may also have gastrointestinal tract involvement where you have nausea and a bit of diarrhea. So that's going to be helpful to the clinician. Also for the clinician to understand sort of what you've got, where you're at, and what to look forward to as far as how the disease is going to progress. The common cold will progress all the way to the top of the period of illness in about 4 days, and it should run its course in 7 days. So this is why when you go to the doctor and you've been diagnosed with something and they say to you, if you're not feeling better in a week, come back and let me know, because if you have a common cold, you should be done with that within a week. If you have those signs and symptoms longer than that, then what might be happening is that you now have a secondary bacterial infection of the upper respiratory tract. Now, if we look on the other hand at influenza, influenza, as we know, is an abrupt acute onset, right? So it comes on really quickly, about a day to peak, and it lasts for about 2 weeks. So it has a much longer duration, and your cough can go for a couple of months after you've actually cleared the infection, right? So it's not an infectious cough. It's a cough that your body is using to just get the extra secretions out of the bottom of your lower respiratory tract. So if you have the flu, the doctor doesn't expect that you'll be feeling better for about 2 weeks. Might not be very good news to you, but that's why they wouldn't be worried if you said, I've been feeling like this for, a week and a half, or if you had all of these symptoms and you said, but I just keep coughing, those are all normal to the disease course. Now, in terms of what they would tell you about how to keep other people in your household and around you from contracting the disease from you, right? Common cold is contact transmission. Contact also vehicle, right? So contact meaning direct, indirect fomite, indirect droplet, but also can be airborne. So that virus can stay airborne for more than a meter, which means that you can contract that a lot. It's a lot more contagious than the flu, though they're both contagious diseases. But it is more because notice the flu, on the other hand, transmission is direct and droplet contact. So within contact, not even indirect, but only droplet and direct contact. So as you go to a clinician and you're wondering why they're asking all these questions and what they're aiming to figure out from all of that information, this is sort of a diagnostic for you. Now, if you have the lecture notes in front of you, this is not in there because I don't expect you to memorize any of the details of these. The only reason I'm showing you this is so that you have an idea of how occurrence, severity, extent of host involvement, progression, and transmission might be helpful in diagnosing, treating, and preventing the spread of particular diseases. Our last topic for chapter 14 is one I think that is very important to all of you, and that is the concept and the topic of healthcare associated infections, which is a new name for what we in the past have called nosocomial infections. So you might be thinking, never heard of a healthcare associated infection, but hopefully nosocomial infection sounds more familiar to you. Now, nosocomial or healthcare associated infections, it's important to understand, happen as a result of being hospitalized or put into some kind of a care facility. These are not infections that are present prior to a person coming into the hospital or healthcare environment that get worse. These are things that people come in without and contract while they are in a hospital or healthcare setting. And they affect about 15% of all hospital patients. So these are a big deal. And the reason why we talk about them in general microbiology is because you are a big part of how healthcare associated infections happen and how healthcare associated infections can be stopped. And the reason for that is in this diagram in the upper right hand corner of the overhead, right? Notice microorganisms are in a healthcare associated environment. Right now, that is clean as we try to be, that's always going to be the case. And who is in the hospital? People who are sick, right? So we've got microorganisms, we've got compromised hosts, and then to make that a perfect storm, we have nurses, we have physicians assistants, we have nurse practitioners, we have doctors, we have respiratory therapists, occupational therapists, physical therapists, phlebotomists, food service people, janitorial staff, all of those people moving from room to room, which means that you guys accomplish the third side of that diagram, and that is the chain of transmission. So a compromised host cannot get sick if they never come into contact with the microorganism, but oftentimes it's the healthcare worker or the person working in the hospital or healthcare setting who is moving the microorganism to the compromised host. So healthcare associated infections primarily affect the upper respiratory tract, surgical sites, lower respiratory tract, and gastrointestinal tract, right? So if you look at this pie chart at the upper left-hand side, we see that the most of these infections are either lower respiratory tract infections, which are ventilator-associated pneumonias, right? So these are pneumonias that are contracted because a person is intubated. The same number, 22% of these infections are surgical site infections. And then if you look above those two numbers, we have 17% are gastrointestinal infections, which happen when people are taking antibiotics, which rid them of their normal flora, and then they become colonized by hospital pathogens like Clostridium difficile and things like that. On the other side, you see that orange pie piece, 13% are catheter-associated urinary tract infections. 10% of healthcare-associated infections are catheter-associated blood infections. So these are central line and IV associated septicemias. And then about 16% of them are in other areas of the body. So urinary tract infections, surgical sites, lower respiratory tract infections, and gastrointestinal infections, which amount to, in the United States, 2 million infections and 70,000 deaths per year. And those are huge numbers, especially when we consider the fact that since the year 2000, there has been an overall increase in nosocomial or healthcare associated infections of 36%. Now, the bottom line is that nosocomial and healthcare associated infections are expensive. They cost between $4.5 and $11 billion per year, depending on the person who's doing the study. But even $4.5 billion a year is enough money to really get people thinking about why these things are happening and how to stop them from happening. So there have been tons of studies done, lots and lots of studies done on how to drop that number, right? Not only to stop this increase, but to get those 2 million infections down to as small a number as possible. And here's the kicker. Of the 3 most common kinds of hospital-acquired and nosocomial infections, which include ventilator-associated pneumonias, central venous catheter-associated bloodstream infections, and catheter-associated urinary tract infections, the best thing that you can do is wash your hands between patients and between procedures. Now, the crazy thing about these standards, right, this is a meta study where they've gone through and looked at lots and lots of different studies, right? And these are evidence-based guidelines for the prevention of hospital-acquired infections. What this means is the majority of people who are caring for patients are not following effective hand hygiene procedures. They are not washing their hands for 30 seconds between patients with soap and water. They're either not washing their hands, they're washing them for too short a period of time, or they're foregoing the hand washing in lieu of, say, an antibiotic foam or something like that as they walk into and out of patient rooms. And what the studies say, what the evidence say, is that if you wash your hands between patients, you will drastically reduce the number of hospital-acquired and nosocomial infections. And if you don't get anything else from this class but that, then that's fine. I mean, once you're done and you've earned your A, I don't care, right? You can forget all of it, but when you get into the hospital, remember that you got to wash your hands, right? Because these are sick people and they're already sick. They don't need to be dealing with an additional infection that is caused because someone has been too busy or too lazy or too flippant to wash their hands between patients. Alrighty, well, that's it for chapter 14. Sorry, I know I'm not the best at talking over the presentation, but hopefully that gets the point across. Obviously, like all the other chapters, we've got the chapter outline you can look at. We've got animations and things that you can look at that make some of these concepts more clear. You've got the online quizzes, and as always, anything that's in the textbook that you don't find on the quizzes, the outlines, or the lecture notes will not be on your test.